Good morning, everyone. I'm glad to have this opportunity to present my recent research work here. My name is Qiujing, a professor from Tianjin Medical University of China. Today, my topic is on serine metabolism and antiviral innate immunity. We all know that when viruses, bacteria, and other infectious agents invade mammals, the body will use innate and adaptive immunity to fight them. Type 1 interferon, mainly include interferon alpha and interferon beta, plays an important role in antiviral innate immunity. Host cells recognize viral RNA and DNA through different kinds of pattern recognition receptors, including RIG-I-like receptors and SGOC. PRRS then recruit adapter proteins such as mouse, SLIM to activate TBK1 and or IKK kinases, TBK1, IKK phosphorase IF3, and NF-κB to induce their nuclear translocation and subsequent transcription of type 1 interferon and inflammatory cytokines. Upon binding to interferon receptors, type 1 interferons activates JAK-STAT1 signaling and promote the expression of interferon-stimulated genes to counter virus infection and coordinate adaptive immunity. Glycolysis product, lactate, prostaglandin, PGE2, or metabolites, has been reported to impact type 1 interferon production. However, whether and how other metabolism participate in antiviral innate immunity is largely unknown. We are specifically interested in serine glycine 1-carbon metabolism network. Serine is a non-essential amino acid. It can either be synthesized, de novo synthesized, or be taken up by transporters from the microenvironment. During de novo synthesis, glycolysis-derived 3-PG can ultimately be converted to serine through a series of enzymatic reactions whose first key step is catalyzed by PHGDH. Serine-derived 1-carbon metabolites, including S-adenosine, methionine, SAM, glutathione, and nucleotide, plays critical roles in macrophage inflammation and T effector expansion. However, whether SGOC metabolism is involved in antiviral innate immunity remains largely unknown. By using qPCR, our initial experiments showed a broad downregulation of the SGOC network gene products in mouse primary peritoneal macrophages at 5 to 10 hours post-infection with the RNA virus, Sendai virus. Among these pathways, genes involved in the serine synthesis pathway and coding exogenous serine transporters were prominently expressed by cell. By using LC mass, we found that SGOC metabolites serine, glycine, SAM, and SH decreased significantly 6 hours after cell infection with BMDMs, whereas methionine and GSH did not change significantly. In summary, these data collectively demonstrate that downregulation of SSP, serine transporter, and serine metabolism acts as a hallmark of virus-infected cells. Further study shows that type 1 interferon signaling was sufficient and at least partially mediated this reduction. To investigate whether PHGDH has a role in antiviral innate immunity, we generated myeloid conditional knockout PHGDH mice through LOX-P lysis-2-Cre system and found that knockout mice had normal differentiation and numbers of PMs compared with their counterparts. Next, we treated wild-type and knockout peritoneal macrophage with SEB, and we found that the secretion of interleukin-1 beta protein and the phosphorylation of TBK1-alpha-3 was significantly upregulated in knockout macrophages stimulated with SEB relatively to those in macrophages. Moreover, we found that although PHGDH expression substantially reduced the upregulation of VSV-induced interferon beta mRNA expression and restored the VSV GP fluorescence in PHGDH knockout cells, PHGDH dead enzyme mutants did not. These results indicate that PHGDH enzymatic activity is required to inhibit VSV replication. We further found that inhibition of exogenous serine transporter enhanced cell-induced interferon beta expression. The combined depletion of serine and glycine resulted in increased interferon beta M1 mRNA. These results indicate that serine metabolism inhibits interferon beta-mediated cellular antiviral activity. Serine-restricted mice showed significantly enhanced interferon beta M1 RNA, less VSV copies in the liver, and reduced lung injury, and survived longer than control-fed mice after virus infection. Inhibition of serine de novo synthesis by using PHGDH knockout mice or PHGDH inhibitor protects mice against viral infection. Overall, these findings indicate that inhibiting serine metabolism enhances antiviral innate responses in vivo. Taken together, these results indicate that serine metabolism inhibits antiviral innate immunity in vitro and in vivo. This effect is through TBK1-alpha-3 axis. We then explored which SG-derived metabolites may be responsible for inhibiting antiviral innate responses. Interestingly, we found that in the absence of exogenous serine, the addition of SAM, but not GSH or formate, significantly lowered the increase of interferon beta M1 RNA to the levels seen in the control cells. We further found that addition of SAM significantly reduced cell-induced interferon beta mRNA expression. Furthermore, SAM restored the interferon beta mRNA of knockout PMs to levels seen in wild-type PMs after virus infection. To further determine the dynamics of serine metabolism in relation to SGOC metabolism, we used a U13-carbon glucose tracer and found that virus infection significantly reduced isotope-labeled glucose incorporation into serine and SAM. Moreover, we found that PHGDH knockout PMs showed significantly reduced intracellular SAM levels of SAM and reduced the labeling fraction of glucose-derived serine and SAM. Taken together, our results show that virus infection and pH knockout reduced total SAM and SSP-derived cell. We next tried to elucidate the underlying mechanism. By using RNA-seq, we found that PHGDH knockout BMDMs and PHGDH knockdown law 264.7 cells had common changes in the expression levels of 25 G genes compared with their corresponding controls. PHGDH pathway enrichment analysis of differentially expressed genes showed dramatic enrichment of lysosome-related genes in PHGDH knockout BMDMs. Then two lysosome-related genes were selected for the analysis of virus replication, only knocking down ATP6-winning-D2 alone by siRNA in BMDMs significantly downregulated virus-induced interferon beta M1 RNA and upregulated VSV replication. D2 is a subunit of vascular ATPase, where ATPase is an ATP-dependent proton pump consisting of peripheral V1 domain that hydrolyzes ATP and integral domain that translocates protons in a variety of cellular membranes to acidify intracellular components, including endosomes and lysosomes, where ATPase has been implicated in regulating multiple bioprocesses, including protein degradation or facilitation of virus fusion. However, the role of VATPase in antiviral immunity is currently unknown. Interestingly, we found that PHGDH deficiency upregulated ATP6-winning-D2 transcription, and exogenous SAM treatment could rescue this effect. SAM is a major methyl domain for histone methylation, which plays critical roles in chromatin status and gene transcription. Therefore, we asked whether changes in the histone modification state occurred when cells were depleted of serine. We found that PHGDH deficiency increases the expression of ATP6-winning-D2 by inhibiting SAM-dependent H3K27ME3, and exogenous SAM treatment could rescue this effect. Together, all these results suggest SG deficiency increases the expression of ATP6-winning-D2 by inhibiting SAM-dependent H3K27ME3. Next, we raise the question, how does ATP6-winning-D2 regulate interferon beta production? Our results show that knockdown of ATP6-winning-D2 significantly upregulated VSV replication and inhibited interferon beta mRNA. To address whether ATP6-winning-D2 can protect mice from virus infection, we challenged ATP6-winning-D2 knockout mice and wild-type mice with VSV virus. ATP6-winning-D2 knockout mice showed more lung damage, less interferon beta M1 RNA, and enhanced VSV RNA replication in the liver. These data indicate that ATP6-winning-D2 is required for interferon beta-mediated antiviral innate immunity in vitro and in vivo. Moreover, the enhanced transcription of interferon beta-1 mRNA and phosphorylation of TBK1 in PHGDH knockout PMs induced by cell infection were at least partially reversed by knockdown of ATP6-winning-D2, indicating that other proteins such as other subunits of VATPase may be involved. We next wondered how ATP6-winning-D2 regulated interferon beta production. Recent studies showed that upon virus infection, YAP is targeted for lysosome degradation by IKK epsilon-mediated phosphorylation, and YAP negatively regulates interferon beta-mediated antiviral innate immunity by inhibiting F3 or inhibiting TKB1 activation. Therefore, we asked whether ATP6-winning-D2 could influence YAP expression. We found that ATP6-winning-D2 overexpression and virus infection robustly reduced YAP protein levels. Knockdown of YAP robustly restored ATP6-winning D2. Deficiency-induced decrease of interferon beta mRNA expression. Overexpression of YAP significantly reduced cell-induced interferon beta-1 mRNA expression and TPK1 phosphorylation in cells with SG starvation. Taken together, our findings reveal that ATP6-winning D2 promotes YAP lysosome degradation to enhance interferon beta production. Here, our study reveals a critical role of serine metabolism in blocking antiviral innate immunity and suggests that manipulation of serine metabolism may be a therapeutic strategy against viral infection. This work is published on Cell Metabolism this year. This preview in the same issue highly evaluated our work and commented that we have identified serine metabolism as a central integration hub of cellular metabolism, antiviral immunity, and epigenetic regulation. Finally, I thank all my lab members, especially the first author, Longsen, my collaborators, and my grants to support this work. Thank you for listening. That's all.